Hello, I'm Nicola Bordin, a research fellow at University College London. I'd like to thank you for the opportunity to present my work titled Pruning the Protein Jungle, Recent Developments in the CATH Function Prediction Pipeline. One of the main protein resources, UniProt, has already reached 180 million sequences, and there is currently a massive expansion in sequencing data coming in from different sources. There are especially two ongoing projects that produce vast amounts of protein data. Magnify, for bacterial metagenomes, already has 1.3 billion sequences, and the Darwin Tree of Life project, which is aiming for the sequencing and characterization for all 60,000 eukaryotes in the British Islands archipelago, is building up data too. Combined, these two projects amount already to 4 billion sequences, and it makes it really hard to organize and characterize properly. Our data resource, CATH, prunes this protein jungle by classifying evolutionary families of protein domains into structural superfamilies. We expand our knowledge of structural superfamilies with sequence data. We initially use structural data because it's easy to characterize and because it's conserved. We then pull sequences into the structural family, we extract a sequence profile, and then we encode it in a hidden Markov model. Although the structure is conserved, there is enough sequence divergence in the paralogs, and the presence of different functions in a structural superfamily, or as well as we call them, different functional families or fanfams. Fanfams are therefore functionally coherent subsets of sequences within a superfamily we extract and then characterize. So, for the next 10 minutes, I will explain how we generate fanfams, some new algorithms we developed, how we benchmark them, and an application on biological data. So we start by generating these functional families by gathering all the sequences for a given superfamily. We collected each gene ontology term associated to these sequences, and then we cluster the sequences at 90% sequence similarity. From then, we extract just the clusters with at least one experimental Go term, and we encode their sequence patterns in a hidden Markov model. Once we obtain the hidden Markov models, we create a tree of relationship between these. Once we obtain this tree, we cut the tree in order to obtain functionally pure alignments. And this cut is performed by a tool we call Fanfarmer. Fanfarmer looks at the presence of two different kinds of residues in our alignments. There are certain residues that are in conserved positions and are all the same for all the sequences, since they're really important for folding. And then there are others that we call specificity determining positions that are important to distinguish function. And we perform the cut at the boundaries between them. There is a caveat with this approach, though, because it doesn't work on massive trees with thousands of starting clusters and millions of sequences. And there are already 12 massive superfamilies in CATH that contain around 30% of all the sequences. For previous fanfams generation steps, this will take around 6 months in UCL computing time, HPC computing time. And since now they can contain millions of sequences, the current method simply doesn't work. So a way to approach this issue is try to split the initial starting clusters according to some criteria. And one of the best ways to approach this is by looking at the presence of multi-domain architectures, partitions, context. A domain context in a protein usually changes with function. And if we split these initial starting clusters according to this multi-domain architecture partitions, we are able to speed up this process of building and cutting this tree from 6 months down to 6 weeks. And this is how it works. So we gather the starting clusters for the whole superfamily as we did before, but we then generate a tree for each one of the multi-domain architectures within that superfamily. And once we obtain the tree, we cut the tree and obtain the fanfams in the same fashion we used to do before. We gather these intermediary FAMFAMs or starting clusters for a second iteration of the algorithm where we pool these starting clusters and we generate a secondary tree containing all of them at the same time. We then cut the tree again, and this gives us the final FAMFAMs. This allows us, though, to have an n-fold speed up compared to the previous algorithm, depending on the number of MDAs that we have within the superfamily. And this allows us also to run multiple rounds of the algorithm in order to increase the accuracy. Once we obtain these new sets of FAMFAMs, we wanted to test what's their quality. And in order to do that, we used the CAFA benchmark for protein function prediction. CAFA is usually assessed using experimentally characterized Go terms and on a set of 100,000 sequences that different teams need to characterize. CATH, in past editions, ranked in the top 10 for certain metrics, and we wanted to test our approach by seeing how well it would perform compared to our previous method. So in this case, for the next 3 slides, I'm going to show you 3 plots, 2 plots for all 3 metrics for all 3 ontologies on Go. And on the left, you have the misinformation remaining uncertainty plot, where we'd like to have the line to be as close as possible to the axis origin. And on the right, you have a more typical precision recall plot. And from all 3 ontologies, you can see that on average, the new method with MDAs outperforms the old method. So once we realized that our data was sound, we tried testing it on a biological problem. The data set we used for this is the protein Kinome. It's an important set of proteins. Most of them are dark targets, and from a structural standpoint, they consist of 2 lobes connected by a hinge region. So the current, another reason why we wanted to update the protein Kinome is that the current golden standard in protein kinase classification, called the Manning or Kinbase classification, it hasn't been updated since 2012. It contains mostly model organisms and it's manually created. So since then, there's been hundreds of thousands of sequences that have been deposited but not classified in Kinbase. The first test, it was to encode a protein kinase using CATH domains. So we noticed that the N lobe corresponds to a specific CATH domain, and a C lobe corresponds to another CATH domain. So we gathered in our database all the sequences that match this criteria, where we have also a hinge region of around 20 amino acids in between. And this corresponds to around 230,000 sequences. We then tried to classify using into FAMFAMs, or in this case, how we call them, KIMFAMs. And we noticed that we have a 17-fold expansion in sequences classified. And what's interesting is that within our KIMFAMs, we contain also the original Manning classification. And on top of that, we have a massive expansion, especially in bacteria. So once we gathered this data set, we wanted to test how well we classified these protein kinases. And for that, we used the enzyme classification purity test. Enzyme classification is the EC numbers consists of 4 sets of digits, where the first 3 classifies a protein according to its chemistry, and the 4th one describes the substrate specificity. So in the case of FAMFAMs, we would expect to have a single function within it, and just a unique EC term associated to it. So once we tested the EC purity at the EC3 level for between KIMFAMs and Kinbase, we noticed that we have comparable accuracy for chemistry with a slightly higher purity for our KIMFAMs, but what we were mostly interested in is the purity at the EC4 level, because it's usually tricky with automated methods to detect this substrate specificity just from sequence. So, and we noticed that we have an actual higher purity compared to Kinbase. I'd like to stress the fact that this is a completely automated method compared to something that was built over the years through manual curation. So the next step for this is going to be to try to look at drug repurposing and potentially investigate novel kinases that we've discovered in bacteria and other organisms. And I'd like to conclude the last portion of my talk with a machine learning approach that a colleague of us applied on Schistosaccharomyces pombi to see if information encoded in FAMFAMs can improve a network-based machine learning approach. So Harris Coles in our group started by gathering for the pombi proteome all string network data associated to it, with the exception of text binding. And he was able to reduce the number, these 30,000 features that were present in these networks down to 256 dimensions. These were mostly highly informative. And we wanted to include FAMFAM data to see if we were able to add an extra layer of information to it. So he started by scanning all pombi proteins against our FAMFAMs, but he noticed immediately that if he wanted to include all the FAMFAM information in this model, it made it almost impossible for his autoencoder to work. So his approach was just to select just the FAMFAMs that contain Go terms associated to pombi sequences, and this was able to make the model work by reducing these 60,000 HMM e-values that he was using down to something much more manageable, that they were around 100. He then noticed that having this extra layer of FAMFAM information on top of his network model was actually improving the information he was able to obtain. By looking at this plot, you can see how, in this case, network data clearly outperforms FAMFAM's data. But I'd like to remind you that FAMFAMs are organism agnostic, while this method and the underlying network data that was used was manually created and custom built just for this organism. And in this case, FAMFAMs are still improving the annotation with information that isn't included in the network data. And this leads me to my final thought about the advantages of functional benchmarking like CAFA. We expect this network data to outperform FAMFAMs on model organisms, and CAFA will be valuable to determine to what extent this is the case for all model organisms. And also it's essential for showing us what are the current trends and help us steer our research towards more promising techniques. And with this, I'd like to acknowledge everyone in our group, and I'd like to thank you for your attention.